think I'm streaming now. I wonder if someone can tell me if I am. I don't know how to see the chat. Yay! I'm actually streaming. I made it work. Thank you. Excellent. Can you hear me okay? Hi, Valerie. Hi, Christy. Can you hear okay, or is there an echo like has happened before? Excellent. Thank you. Well, I set for 12:50 because I wanted to be sure. Good. Thank you. I wanted to be sure we had enough time to get everything set up, so we have a few minutes to make sure everything is running okay. I hope my computer doesn't overheat in all this, but I'm sure we'll be fine. Excellent. So how are things in your part of the country? You're hearing me twice. How about now? Is it still twice? What if I stay very still in the middle and don't move around? I think I'm speaking into my... Okay. Oh, good. Okay. I am just using my computer's microphone. Excellent. Oh, good, Valerie. Thank you. Now I just had to remember the details of meiosis and we'll be fine. We're just waiting until 1 o'clock Eastern time to go ahead and begin. Thank you all for tuning in. Oh dear, what does that mean? A request for access. Ah, I think I know. I'm sorry. Hang on, I think I can do that. I apologize. Thank you for reminding me. Vivian, does that help? Did I get it right? Excellent, and thank you for reminding me. I do that with my students a lot too, it turns out. They're reminding me to open things up to share. It's all good. Okay, just a couple more minutes and we'll get started. Give people a chance to get here at 1 o'clock. All right, well, let's get started. It's about 1 o'clock Eastern Time, and on Saturday, April 18th, good afternoon. Welcome, and thank you for joining me as we review Unit 5, Heredity, looking at meiosis, Mendelian and non-Mendelian inheritance in preparation for the AP Biology exam. My name is Phyllis Robinson. I teach at St. Andrew's Episcopal School in Potomac, Maryland, and I am delighted to be with you today. Linked below this video, you'll find the slide deck from this presentation. You'll find the schedule of the remaining review sessions, and there's still a question submission form for the remaining review sessions. So submit a week ahead of time to maximize the possibility of your question being answered in the session. So my suggestion on how to use this video, it's to refresh your memory of the content from this unit. It's to consider how the material from Unit 5 might be incorporated into this year's AP exam. And it's meant to support, not fully replace, your own in-depth review of the Unit 5 material. Not all topics can be covered here in depth, but I really hope to help answer some of your questions and to just refresh your memory on these things. Do ask questions in the chat. I've got some wonderful moderators who are happy to help answer them as well. If I'm able to see it and answer, I'll do the same. But moderators, I'm counting on you to help field those questions for me. The AP questions on the exam are always about content plus skills. So you might review the AP Biology exam practices that are linked below. And mental health is more important than exam prep. So take care of yourself. You want to sleep the night before and eat well and all that. Those things are important. Your chat moderators are Clifford Nefrata, Christy Martin Philippe, and Valerie Pierce. And please thank them for volunteering their time. I'm really thrilled that they are helping me today. Just a reminder that it's likely that I'll make a mistake somewhere along here, and so please catch me and help me fix that. I also have a cat who sometimes wanders through yowling loudly, and I hope she won't, but I apologize in advance if she does. So it's a lot of material. Let's dive right on in. In Unit 5, there are 3 main ideas, or sometimes called enduring understandings. One is that the heritable information provides for the continuity of life. And so we're thinking meiosis. A second one is that organisms are linked by common lines of descent, from a common ancestry, as you can see in my very tiny little phylogenetic tree here. And that naturally occurring diversity among and between components within biological systems affects the interactions with the environment. So that as sort of a preview. 6 topics in Unit 5, meiosis and genetic diversity, both Mendelian and non-Mendelian genetics, the environmental effect on phenotype, and chromosomal inheritance itself. So topic 5.1, meiosis. I find with my students that this is a complex and confusing topic. It often takes several go-rounds to really understand what's happening, especially because it's so similar to mitosis. So here we just want you to be able to explain how meiosis results in the transmission of chromosomes from one generation to the next. So what we're thinking about is making gametes. That's the point of meiosis. If you start with an egg cell in an ovary, a normal full complement of chromosomes egg cell, the oocyte, go through meiosis, you'll get an egg cell with half the chromosomes. Same thing for the sperm cell. Start with a spermatocyte in the testes, go through meiosis, reduce the number of chromosomes by half, you have a sperm. Join that egg and that sperm in the process of fertilization, and now you're back to a full set of chromosomes in a cell we would call a zygote. And that zygote then goes through mitosis many, many times to become eventually you, or whatever organism, of course, it's going to be. So to back up just a second, that whole half chromosome and full chromosome complement, what's going on there? Think of a normal human karyotype. This is just a picture, and it's an artist's picture rather than a photograph, of all the chromosomes in a single human cell. And every human cell would have all of these chromosomes. Each chromosome has a partner. They come in homologous pairs, and you can see they go from large ones all the way down to small ones. And then finally our sex chromosomes here, which I'll talk about with my non-Mendelian genetics. So this is a full set of a full complement of chromosomes, 2 sets. So basically I've got 2 of every pair. I call this diploid. So over here, my diploid, 2 copies of each chromosome. This particular cell has only 3 pairs of chromosomes, just 6 chromosomes, but the same idea applies as humans. You might notice that I've got pink ones, but they're different shades. I've got blue ones, but they're different shades. And green ones, but they're different shades. And the different shades are showing that one was inherited from the mom and one from the dad. There's a maternal and a paternal chromosome. So that's a diploid. When you go to make the gametes, the eggs and sperm, you're going to want them to be haploid. You want only 1 from each pair, because when that haploid sperm meets up with that haploid egg, you then have a diploid cell, you're back to full. If you didn't reduce the chromosome number, you'd have a sperm with 6 chromosomes and an egg with 6 chromosomes and then a zygote with 12 chromosomes, and that rapidly gets out of hand. So that's our haploid versus diploid, really fundamental to understanding meiosis. And then sexual reproduction is just as, of course, taking that haploid sperm, that haploid egg, making your diploid zygote. 3 chromosomes, 3 chromosomes, 6 chromosomes. So one of the things that they really want you to know is, how does meiosis produce haploid cells? They're not interested in the details. We don't really expect you to go through the all the different steps of meiosis and what happens in anaphase 1 versus metaphase 2. We really just want you to understand a couple main things about this. First, that after replication, this is a late interphase cell where replication has already occurred, and we go through the process of meiosis 1, line up our chromosomes, split them, line them up again, split them again. So we have 2 rounds of divisions, meiosis 1 and meiosis 2. Now the reduction, the actual chromosome reduction, occurs in meiosis 1, where the diploid cells become haploid because you're splitting the number of chromosomes in half. Each chromosome is made up of 2 sister chromatids, which is where sometimes we get a little confused with the math. But if you want to just count the centromeres, then you'd see I've got 4 centromeres here in my diploid cell, 2 centromeres here in my haploid cell. Two here in my haploid cell, and then 2 down here in my final haploid cell, and I've lost my sister chromatid at this point. The main parts of this that are crucial for understanding how this happens is here in metaphase 1, the homologous chromosomes will line up double file. They line up beside each other so that they can then separate to opposite sides of the cell. If you think back to mitosis, they did not have to line up side by side. All the chromosomes just lined up along the middle, and the sister chromatids split. Nope. Here we need to make sure we get one of chromosome number 1 and one of chromosome number 2 going each way. And then down in metaphase 2, they'll line up again as normal, like just along the metaphase plate. And during anaphase 2, the sister chromatids now separate, telophase 2, cytokinesis, and you end up with your 4 cells. So how does meiosis produce haploid cells? You got to be thinking about how they line up in metaphase 1, and then in metaphase 2, followed by the splitting in anaphase. So meiosis, best way to really get meiosis is to actually act it out with string or pop beads or clay or something on a table. It makes the most sense when you can actually take chromosomes, replicate them, and then move them about until you have 4 haploid cells. So I hope you've done that in your class, or that you get a chance to do that at some point here. I think that makes a big difference. So to summarize, before division, we've got 1 diploid parent cell. We replicate it. Meiosis 1, we split the pairs. Meiosis 2, we split the sister chromatids. Cool. So one of the things they ask us to be able to do is to describe the similarities and differences between the phases and outcomes of mitosis and meiosis. I've got a diagram over here that kind of outlines that a little bit. Similarities, well, they all go through prophase, metaphase, anaphase, telophase, cytokinesis, although we don't need to know the actual specific details of those phases anymore. It's sort of a general process. But they both go through that. They both, mitosis and meiosis, what's important is the movement of the chromosomes and the separation into cells. And you got to remember to replicate your chromosomes first. Start here with a parent cell, replicate them like you see here, line them up, divide them, boom, you've done mitosis. A little bit more complicated for meiosis. So some of the differences, though, are pretty obvious in this diagram. First of all, the number of daughter cells you get. In mitosis, you end up with 2 daughter cells, and they are identical, not only to each other, but to the original parent cell. You're trying to make exact copies. If you're growing an embryo, you want all the copies to have the same genetic complement as that organism grows up. In meiosis, you can see you get 4 daughter cells, and each of them has only half the chromosomes that the parent cell had. And that, as we explained, is so that it can match up with the other gamete and produce a diploid zygote. The number of divisions is different. 1 for mitosis, 2 for meiosis. What lines up during metaphase is different. So in metaphase, chromosomes line up just in an orderly file. In mitosis, first the homologous pairs, and then the remaining chromosomes will line up. And the crossing over is the 5th thing that's different. I'm going to explain that a little bit more in detail later. It occurs in meiosis during prophase 1 early in the process. It does not occur during mitosis. So to summarize, Here's a chart that you can summarize, and you can look through this to look at the number of divisions. Does independent assortment happen? Synapsis is another name for when the chromosomes come together to do crossing over. What the number of cells are in your outcome, diploid versus haploid, what it's used for, body cells versus the gametes, and the genetics. Are there identical cells to the parent and to each other, or there's variation? Here's a free response question from a few years ago that took some of this content into account. Both mitosis and meiosis are forms of cell division that produce daughter cells containing genetic information from the parent cell. They ask you to describe two events that are common to both mitosis and meiosis, and they ask you to figure out how the process of cell division leads to the differences when you get meiosis. All right? So, next topic. How does meiosis produce genetic diversity? I'm going to explain how the process of meiosis generates genetic diversity. As a quick moment of background, this is a hydra. You may have gotten to see a hydra during your biology class. I hope so. They're cool little creatures. They're cnidarians, very, very small. They also they do sexual reproduction, so they produce eggs and sperm. But a hydra can start producing cells on some part of its body that then grows into a whole new hydra, basically a clone of the parent. That new hydra will then eventually grow big enough, drop off, and become a separate individual. Cool, I made a baby, but it's genetically identical to the parent. Nothing particularly wrong with that. And in fact, you can increase a population rapidly if you're relying on cloning or budding to make a lot of offspring. But you don't get much genetic diversity when you do this. And genetic diversity is what evolution is dependent on. Organisms can change throughout, populations can change as the environment changes if there's genetic variation. So this is an example of not genetic variation. Three main ways that meiosis ensures genetic variation and sexual reproduction ensures genetic variation. In crossing over, this happens during prophase 1 of meiosis. The homologous pairs actually kind of group together and hug. They join themselves together, and then they swap pieces of DNA. This particular example shows them swapping just two pieces down here. So my blue parental one and my red blue paternal one and my red maternal one now just swap ends of a chromosome arm, so that my blue paternal is now carrying some of the DNA from the red maternal, and vice versa. In real life, many bits of DNA swap, and it's pretty random as to what swaps where. So you, again, increase some genetic diversity through crossing over. If you compare a diagram of crossing over and not crossing over, you can see the distinction a little bit better. Paternal chromosome, maternal chromosome, and each of them has a slightly different colored tip, so you can kind of track their progress. Here they've replicated. If there's no crossing over, you go through meiosis 1 and meiosis 2, the paternal chromosomes retain their little green tips. The maternal chromosomes retain their little pink tips. And that's fine, it's just less diverse. Then if you do have crossing over here, you'd have some swapping. Again, go through meiosis 1, go through meiosis 2, and now the paternal chromosomes, one has the original tip, and one has some of the maternal DNA instead. And the same thing over here, the maternal one has a new paternal bit of DNA, and it's original. So these four are now all slightly different from each other, whereas over here I've only got two possible differences. So that's the power of crossing over. And again, it's not just one tip. This is happening quite a bit along each pair, and we have 23 pairs doing this, so really the possibilities are practically endless. All right, a second way that meiosis generates genetic diversity is through random fertilization. An ovum, an egg, could be fertilized by any of one of many different sperm, and each of those is carrying a slightly different complement of chromosomes, and it's random as to which one will match up with the egg. And in organisms that are producing a lot of eggs, you now have an even greater possibility for matching up different sperms and different eggs. So random fertilization is the second way meiosis and sexual reproduction generate genetic diversity. And the third way is through independent assortment. So haploid gametes will have a mix of maternal and paternal chromosomes simply based on how the different chromosomes assort themselves during the process of meiosis 1 to meiosis 2. So here I've got two blue paternal, and they both end up going into the cell, and so my gametes have both paternal chromosomes and both maternal chromosomes. Over here, where they could just rearrange themselves in the cell, I've got a paternal chromosome with a maternal chromosome, so they now have a little bit of mixing and matching going on down here. Altogether, I've got 4 different ways these 4 chromosomes could have assorted themselves randomly and independently into the gametes. And of course, you now have 23 pairs of chromosomes for humans, and you've got crossing over going on. So you don't just have 4 combinations, you really, again, have an incredible number. There is an exclusion statement in our course and exam description. The details of sexual reproduction cycles in various plants and animals are beyond the scope of this course. What that means, if you've studied alternation of generations in, say, ferns or funguses, that won't be included on the exam. Nice to know, but it won't be part of the exam. All right, let's move on to Mendelian genetics. We're going to first explain how shared, conserved fundamental processes and features support the concept of common ancestry for all organisms. I've included a simple family tree of all the organisms. We've got bacteria, archaea, and the eukaryotes, the 3 domains of life. Here's the mammals. We are pretty recently evolved, and we had a common ancestor back here somewhere with fish and birds and reptiles. Trace us a little further back, we've got some common ancestors with other animals, common ancestors with funguses and plants. Ooh, back here, a common ancestor with the archaea. Keep on moving back, and there's a common ancestor even with the bacteria. So we all evolved from some kind of common ancestor. And there are some parts of our cells and our metabolic processes that still are the same throughout all these organisms, which lends evidence to the fact that we've come from a common ancestor. So one of those pieces of evidence is that the DNA and the RNA that exist in all of us, they are the molecules that carry genetic information in all life forms. So prokaryotic DNA, bacterial DNA, is a loop. It's a single chromosome. It's found in a loop. And that eukaryotic DNA is linear. It comes in straight lines or curvy lines if they're all muddled together. I think there's 3 chromosomes in this picture altogether. But regardless of how it looks or how big it is, they're all made of the same kind of DNA. Adenine, thymine, guanine, cytosine, phosphate groups, the deoxyribose sugar. It evolved early, and all of us have the DNA. The RNA, there's some slight differences between some of the different RNAs, but fundamentally they're extremely similar as well, with ribonucleic acid, single-stranded, adenine, uracil, guanine, and cytosine. Ribosomes are also found in all cells, and we believe that they are also an ancient representation of what's been inherited, what's been conserved and shared throughout time. So a ribosome, you will remember, is a small structure that helps with protein synthesis. It facilitates the movement of the tRNAs, bringing their amino acids in, joining up with the mRNA to create an amino acid chain. This is the process of protein synthesis, fundamental to all cells. So the process, I guess you could invent a bunch of different ways that you could make protein chains, but the same way occurs in all cells, fundamentally. You've got the same type of ribosomes and the same process. So again, that harkens back to our conserved and shared ancestry. And finally, core metabolic pathways, such as glycolysis, are essentially identical in all cells. Start with glucose or some organic molecule and break it apart stepwise, releasing energy to make ATP, removing electrons, which will be reducing our NAD, which we saw in Unit 3, I think it was. So the whole extensive process there. We continue that process, and you end up with pyruvate or some derivative. We all do this. Bacteria, funguses, oak trees, lions, us. We all depend on very similar enzymes. Again, this is a process of that protein synthesis that we all share. Same types of enzymes, break these down. This is basically, think of this as a tried and true pathway. It evolved early, it worked, we all still use it. So I think that is another powerful piece of evidence. And the other thing we want to think about with Mendelian genetics, getting right into the meat of it here, want to explain the inheritance of genes and traits as described by Mendel's laws. A quick vocab lesson, just to back us up a little bit. So here I've got a homologous pair of chromosomes. The DNA in the chromosomes codes for different traits, and we call a sequence of DNA that codes for a particular trait. We'll call that a gene. Now on the homologous pair, they each have a gene for, in this case, flower color. This one has purple, this one has white. So while it's the same flower color gene, it's different variations. And we call the variations of genes the alleles. So genes and alleles are related, but not exactly the same thing. And you probably remember dominant and recessive. So if a dominant gene is present, then the organism will display that phenotype. Purple is dominant over white. So if you have a purple allele, the DNA to actually make purple pigment, then you're going to get purple flowers. The only way to get white flowers is to have both recessive alleles, no purple alleles to overshadow it. A little bit more, the genotype and phenotype are ways we can talk about the organisms. So the genotype is what are the two alleles you have? Are they both homozygous, homozygous dominant? And we'll use capital letters to represent that. Are they heterozygous, where I've got a capital letter and a small letter to represent my dominant and my recessive allele? Or are they homozygous recessive? And I'm going to use two small letters, two lowercase letters, to represent homozygous recessive. And the phenotype that goes along with the genotype. So if I've got a big P in this case, whether it's homozygous dominant or heterozygous, that's my genotype, the genes that are present. The phenotype is what I see, the physical appearance of the organism. So if I've got a capital P, I'm going to have a purple flower, whether it's homozygous dominant or heterozygous. If I've got two little p's, it's homozygous recessive, then I get to be a white flower. So my phenotype is white. I remember phenotype is for the physical trait. Even if you can't see it, even if it's a trait that's an enzyme inside your cell, it's still a thing. So it's the phenotype. Genotype is it the dominant or recessive alleles that you have. All right? And so I think we looked at homozygous and heterozygous there as well. So Punnett squares. My kids love doing Punnett squares because they're practical and they're kind of fun to do, although they can get complicated. We use Punnett squares to predict the probability for the genotypes and phenotypes of the offspring that you might get. So this is a kind of a complicated one to start with, what I wanted to just show you is got a couple of grandparents here. They had a bunch of kittens. Let's mate those two cats. I know let's pretend they're not brother and sister, and that's okay. And so what you would do is see what possible offspring they could have by doing a very large Punnett square and counting up all the possibilities there could be. And you'll notice that it's not an even split. I've got more that have a purple background, which suggests a short tail and a brown coat. Not so many that have a green background, which is my white coat with a long tail. So it's not, let's back up a little bit. Let's do a simple one. Let's talk about a single trait cross. And I'm going to use flower color as my example. So purple and white are the colors that Mendel used. His pea plants were purple and white flowered, and that's where he figured out some of his traits. So in pea plants, purple is dominant, and we use a big F to represent that here. White is recessive, and we use a little f to represent that here. Now, let's cross two purple parents, and I'm going to assume for the sake of this exercise that both are heterozygous. So they're both big F, little f, which also means this is a monohybrid cross. It's a special kind of single trait cross. What I do is I make a Punnett square and I put here what one parent could donate let's say in his gametes. So he could either donate a big F, so some of his sperm will have big Fs, some will have little f's, about half and half. In the female, about half the eggs will have the dominant allele and half will have the recessive allele. So I've separated what could go into the gametes. That's what goes on the outside of the box. And then you just match up the letters to see what you get. And I've got the genotypes and the phenotypes given here. This is a pretty simple one that most people learn in like introductory biology. you end up with a genotype ratio of 1 to 2 to 1, 1 big F, homozygous dominant, 2 heterozygous, and 1 homozygous recessive. The phenotype ratio, however, because you have 1 big F means it's purple, just like 2 big Fs. I have 3 that are purple, 1 that is white, and so my phenotypic ratio is 3 to 1, purple to white. It's going to be important a little bit later on when we get to some non-Mendelian genetics. So remember, these are just probabilities. If my purple parents happen to produce exactly 4 babies, I'm not guaranteed 3 purples and a white. I could get 4 purples. I could get 4 whites, although that's less likely. These are just probabilities. What, however, if you don't know the genotype? So here's a purple pea plant flower, but looking at it, I can't tell if it's homozygous dominant or heterozygous. Is it big F, or big F, little f? You don't know by looking. The only one I know by looking is the white one, because that has to be little f, little f. It has to be homozygous recessive. So what you do in this case is you do a test cross. And I'm going to change letters on you here because the letter choice actually doesn't matter at all. So that's one reason I chose this one. But what I wanted to show you, the important thing here is we're going to test. We're going to mate our purple, which could be homozygous dominant or could be heterozygous. We're going to mate it with a known genotype. The white has to be little w, little w. If all of our offspring are purple and we get a bunch of offspring, we don't get any whites, we're going to pretty much assume that I've got a homozygous dominant, a true breeding purple flower. If, however, a bunch of the offspring are white and another bunch are purple, could be a 50-50 split. It might not be exactly. But if I've got got a bunch of whites, I've probably got a heterozygous parent. This works for other things. You have a brown mouse. Is it a big A, or big A, little a? Again, you cross it with a white mouse, who's little a, and see what you get for the offspring. If you get some whites, then you probably had a heterozygous. If you get all browns and you have enough to sort of statistically say, yeah, I think I have enough offspring here to guarantee that I probably have big A, then you probably got a homozygous dominant mouse. All right. A 2-trait cross, let's go back to these cats. In this case, the tricky part here is figuring out what to put on the outside. What alleles do I put into these sperms and into these eggs? And really, the trick is pair up each S with each B until you have all the possibilities done. Many people learn to use algebra here. The concept of using the FOIL method, put the first together, outside, inside, last. You may have been taught that by your teacher. So I've got my two, I end up with a big S and a big B, big S, little b, little s, big B, little s, little b. There's no other ways to pair up my S's and my B's. Same thing for the female. And in this case, because this is kind of a special one, they're both cats are dihybrids. I've got 2 parents, dihybrids. This is a dihybrid cross. And this is a famous ratio that you should know. The ratio of the offspring is going to be 9 to 3 to 3 to 1. 9 of our 16 possibilities, that's all the purple ones here, are dominant, show both dominant traits. 3 of the 16, this one, and this one, show 1 dominant and 1 recessive trait. They've got short tail dominant, and then they've got the recessive white coat. These 3 show the recessive long tail, but the dominant brown coat, and only 1 of the 16 shows both recessive traits. Again, this is just probabilities. Let's say these 2 cats have 16 kittens altogether, maybe a couple different litters. They're not guaranteed to have this exact outcome, but these are the probabilities. You can also see a 2-trait cross that's not a dihybrid cross. All you really need to do is figure out the gamete combinations, make your Punnett square, and then count the outcome, which will not be a 9 to 3 to 3 to 1 ratio. That's only for the dihybrid cross. So I just made up an organism. It doesn't matter. Maybe you used a fun organism in your class. Some people do unicorn genetics. Some people do dragon genetics. I just made these up. This could be unicorns. Doesn't matter. Tall is dominant over short. Brown is dominant over white. Here's my parent genotypes, my P generation. And to combine them, I can combine a big A with a big B for 1 gamete. I can combine a big A with a little b for another gamete, and that's it. I don't really have any other possible combinations. If I wanted to put something here and something here, there would just be repeats, which is okay. I'm just going to eliminate that. Same thing down the side. Pair up a little a with a big B and a little a with a little b, and those are my gametes over here. When I do my Punnett square and I count up my results, I will get 3/4 of them should be tall and brown, 1/4 should be tall and white. That's how you do the 2 trait crosses. If you have more than 2 traits, we're going to use the rule of multiplication instead, because frankly, a tri-hybrid cross, I've done it once, it's a nightmare. If you get 27 boxes and you don't want to go there. So here's how you use this. It's a slightly different question too. Given these parents, so these are some parents for a pea plant that has some different traits. The question is, what's the probability of getting an offspring with the genotype of a specific type? In this case, big A, little b, big C, big C. What you do is you take each set of letters independently. So take the A's, big A, times big A, little a, and do a little Punnett square down here. And your probability of getting a big A is 1/2. Okay, hold on to that. Do the same thing for the B's. You'll find the probability of getting a little b is also 1/2 based on these parents. And the probability of getting a big C based on these parents is 1/4. So you take those and the rule of multiplication, you multiply them together. So your chance of getting that offspring is 1/16, or you could divide it out and get the decimal. So that's how I would use. You could actually use this for a 2 trait cross too, if you wanted to find out what an exact offspring is going to be. Okay. So I see some chats going on in the chat, and my moderators are jumping in. Thank you so much for helping. This is complicated stuff. I appreciate your efforts. All right, non-Mendelian genetics. I love this picture because to the untrained eye, it's genetics. I take a father's trait and a mother's trait, I mix them together, and I get the perfect baby. Well, that is not always true. There are deviations to this, and we're going to explain the deviations from Mendel's model of inheritance of traits. There are several different ones to look at. First of all, sex-linked chromosomes. I want to remind you this was our karyotype, and this showed the sex chromosomes down here. If in humans, if you are female, you have 2 X chromosomes. If you're a male, you have an X and a Y. There are some variations on this depending on perhaps different genes or different possible mutations, but by and large, XX is female and XY is male in humans. I want to look at these two a little bit. You can see the X chromosome is a whole lot bigger than the Y chromosome, and as a result carries a lot more genes that the Y chromosome just doesn't carry. You may have studied or done some genetics with Duchenne's muscular dystrophy on the X chromosome, not on the Y. The colorblind gene, red-green colorblind on the X, but not on the Y. Fragile X syndrome, hemophilia is one that we often use as an example. It's on the X chromosome, but not on the Y. The Y does have some genes, not very many, compared to the compared to the X chromosome. So how does this play out when we're actually trying to solve genetics problems? Well, I've got a lot here on this slide for you. Let's look at the genotypes and the phenotypes for a sex-linked red-green colorblindness trait. This is not unusual. Often I have a student who is red-green colorblind. If we look at the genotypes, we're going to write it a little bit differently. We can't just use B for blindness because this is a sex-linked trait, and we have to show that it's attached to the X chromosome. So I've got an X big B, and X big B is a normal female, whereas an X big B Y is a normal male. You don't get to put a B on the Y chromosome because there's just not DNA there. The gene doesn't exist on the Y. And that will be important when we start getting away from dominant. If you're a carrier female, that means you are an X big B, you have normal vision, but you're carrying the allele for red-green color blindness, and you could pass that on to your children. And if you're a female with 2 little b's, X little b, X little b, then you're a colorblind female. On The male side, if you're X little b Y, you're a colorblind male. And you'll notice I don't have anything here for a carrier because the male either has the X big B or the X little b. You don't get that second chance to have another B to perhaps be dominant over a recessive. Okay, so keeping track of all these little X's and B's is part of the trick. So here's a problem that's set up. X big B, X little b, this is a carrier female, and having children with X big B Y, normal vision male. And you do your Punnett square, keeping careful track of your letters. There's a lot of letters to write. But then analyze your results separately. Look at the females and the males independently. So in this case, your offspring, you've got the females up here. Half of them will be carriers, and half of them will be normal. For the males, half of them will be normal, and half will be colorblind. Well, that's again just a prediction, but you don't want to say 1/4 of them will be carriers or something like that. You want to say half of the females will be carriers. So you're being very specific. Okay. Incomplete dominance is another non-Mendelian trait where I've got a red flower and I've got a white flower, and I breed them, I cross them. In Mendelian genetics, if you get a big R, then you get a red flower. But in this case, with incomplete dominance, you get like an in-between trait, a pink flower. And the reason for this is because this particular gene in this flower produces enzymes that will help make a red pigment. The little r does not make that red pigment. It doesn't make a pigment at all. And in many flowers, having a big R means you get enough pigment to make the flower red. In this particular case, it doesn't. You get only some red, enough red to turn this flower pink, but not fully red. So having enough protein, enough enzyme makes a difference here. So here's a homozygous dominant versus a recessive, and we get all pink, all heterozygous. Let's cross a couple of heterozygotes. Now, there's more than one way that these can be, the incomplete dominance is written. Different books choose different ways to write it. This particular one chose using C for flower color, and then said R for red and W for white. That's another perfectly fine notation. Here I've got heterozygous, so I've got pink parents. They've each got a red and a white. My pink parents produce a red flower, 2 pink flowers, and a white. So again, it's 25%, 50%, and 25%. The genotype ratio is very much like the monohybrid cross I did earlier, 1 to 2 to 1. But the phenotype ratio, if this was a monohybrid cross for normal genetics, would be 3 to 1. Here, it's 1 to 2 to 1 again, which is a clue that this is not normal genetics. This is going to be incomplete dominance. Whoa, we could spend like hours talking about these, but I'm going to keep on moving. Thank you for helping out moderators with all the questions. So multiple alleles and co-dominance. Blood type is a great example for both of these. And we're talking about the AB, and O blood types. I'm not going to worry about the positive and negative that we often say. We might say like we're O positive. I'm going to skip the positive and negative for here. Just focus on the ABO blood types. So we have the 4 blood types. The genotype can be written a couple different ways, again, depending on the book. You can write an I with an A to represent the A allele, an I with a B to represent the B allele, and then a little I without anything to represent the O allele because it's recessive. So what happens is the A is dominant over the O. If you have an A and an O, you have A blood type. If you have both A's, you have A blood type. Same thing for B. B is dominant over O, or you can have a homozygous B. Both cases, you get the B blood type. So what happens if you have an A allele and a B allele? Who's dominant over the other? Well, they're co-dominant, which means they have to share. You have both A and B blood type going on. And then if you have both recessive traits, you have the O blood type. And what's really going on over here is type A blood has some kind of protein or glycoprotein marker. It's made a specific enzyme path that creates this marker we're going to call A. B blood type makes a different one. If you have blood type AB, you have the alleles that are making both of these together, and they're both getting put on your red blood cells. Type O, it's not that you have a type O marker, it's that you don't get any of these markers. You just have have red blood cells without A or B markers. What that means, however, is blood type O can be given to any of these because you don't have the proteins on the outside that a person with blood type A would react to. Blood type AB can receive from any of these, but can only give to other ABs. Because if they gave their blood to type A, the type A person would react badly against the type B proteins that are here. So that's a bit about blood transfusions just on the side. Linked genes. There was a question about this that came in earlier, so I want to be sure we touch on this. When Mendel did his crosses, he managed to pick genes that were on different chromosomes, which led to very nice ratios, which we learn with basic Mendelian genetics. When the genes are linked, that means that the two genes are occurring on the same chromosome, and the behavior changes as a result. So in fruit flies, the normal fruit fly is what's called a wild type. And in our case, we're looking at body color and wing type. So the wild type body color is like a tan with some black stripes and normal wings. And then we're going to compare that with a black body or vestigial wings, or a fruit fly that has both a black body and vestigial wings. So the cross here, it's not quite a dihybrid cross. What we're going to do is cross a wild type body with wild type wings. And I'm going to say that I'm going to just determine that the parent is heterozygous. So the wild type big B little b, and the wings are big W little w. So they are carrying the trait for black and vestigial, but they are showing the wild type trait. I'm going to cross that with a fly that is homozygous recessive for both black body type and vestigial wings, little b, little b, little w, little w. I didn't do the Punnett square. You can do the Punnett square, and if you do, you'll find that you get an expected offspring ratio of about 1 to 1 to 1 to 1. And so you would expect, let's say you did this and you counted up a bazillion little fruit flies. This is what Thomas Hunt Morgan did back in the early 20th century and discovered a lot of these lines of transmission as a result. And this is based on one of his studies. Let's say you got 1,600 offspring after doing this. A lot of fruit flies. You would expect 400 of each type. What they got, though, was nothing like that. What they got was different numbers, many more wild type for both, and many more black body vestigial, and not so many of the others. So what happened is you got a whole lot more that showed one parent, the one parent that was wild type, and a whole lot that showed the other parent that showed both recessives, but not so many recombinants in the middle, not so many that combined the trait from one parent, like normal wings and black body, or vestigial wings and a normal body. And you didn't get that 1 to 1 to 1 ratio. What this suggests, when you see this, when you see observed traits that are very different from your expected traits, what's happening, or could easily be happening, is that they're linked. And I wanted to show you this picture of a simple chromosome map of fruit flies. They have 4 pairs of chromosomes, and a lot of most of their genetics has been mapped so we can see what's going on. Look, here's the gene for the black body. Here's the gene for vestigial. Well, that's not true. This is a gene that if the mutation occurs in here, the body turns black. Here's the mutation. If the mutation occurs in the DNA here, you get vestigial wings. And so if the mutation for black body is here and the vestigial wings is here, it's likely that through the process of crossing over, these will end up in the same segment of DNA and not get swapped independently of each other through crossing over. They'll travel together more likely. And that's why you end up with more of those relative to a recombinant. The chromosome doesn't get split here very easily, especially because there's a centromere in the middle. It doesn't get split here very easily during crossing over. And that's what's going on with linked genes. There's a whole lot more we could say about that, but that kind of gives, I hope gives the questioner a question about, an answer about how do you determine if it's linked. There's a practice question, not unlike the one I just showed you, actually. Funny about that, that you can see, and it asks you, it basically says, what's the following, which of the following best explains the observed results of the cross? It explains kind of what I did here. And you can see, look, the expected results are all the same number. The observed phenotypes are not. And the correct answer is that the traits of the body color show. I apologize. I clicked the wrong one. Whoa, this should be B. I will have to go back and fix that. The genes for body color and wing shape are located close to each other on the same chromosome. My mistake for clicking the wrong one, and I will fix that as soon as this is over, and then that PowerPoint slide will be correct. Yeah, mistake. Anyway, moving on to polygenic traits, where you've got 2 or more genes for a single trait. Just sort of be aware of this. We often teach eye color, you're either brown or you're blue. That's not true, and you can look around and note that. There's really at least 2 genes that are responsible for eye color, and depending on what you get and whether it's homozygous or heterozygous, you get a bunch of different types of colors. And again, this is slightly simplified because there's a variety of browns and there's a variety of blues, but it just shows it's more than 1 pair of genes governing that trait. There's more than 1 pair of genes governing skin color. This particular diagram shows 3 genes governing skin color, but I believe it's actually even more than that. It's how much melanin you produce, it's what type of melanin, it's what melanin is deposited in your skin. So there's a lot going on there, all just to produce the trait of skin color. All right, so just be aware that those do happen. And finally, mitochondrial and chloroplast DNA. Mitochondria have their own DNA. Remember, we believe that they came, there's good evidence that they came from bacterial precursors, which have their own DNA. And so mitochondria today still has its own DNA in a circular form, much like the bacterial DNA. And we inherit, in humans, we inherit our mitochondrial DNA from our mothers. Here's why. Here's the egg cell, here's the chromosomes, here's the mitochondria with its mitochondrial DNA. Here comes the sperm with its nucleus. The only thing from the sperm that enters the cell is actually the chromosomes from the nucleus. The mitochondria of the sperm are outside in this tailpiece, providing power to the flagella so it can swim. So the mitochondria here from the father don't enter the egg. So the egg cell has just mitochondrial DNA from the mom, just mitochondria from the mom. It turns out that there are some exceptions to this. There's some evidence that occasionally the paternal mitochondria can get into the cell, but it's a very rare occurrence in humans, as far as I understand. All right, so a couple more topics. Let's see what happens if there's an environmental effect on the phenotype. I want you to be able to explain how the same genotype can result in multiple phenotypes under different environmental conditions. So here are some hydrangeas. Hydrangeas are a commonly planted bush plant with these big pom-pommy flowers. And these 2 flowers are different colors, but the hydrangea has exactly the same genes, whether it's the pink version or the blue version. This is true for 1 or 2 particular species of hydrangeas, not all of them. But in this case, what happens is this. If the hydrangea is growing in slightly alkaline soil, the flowers will be pink. Somehow the genes are activated to make pink, basically make the pink pigments. If the hydrangeas are growing in acidic soil, they will tend to be more purpley, lavendery, or even as deep as the blue, which apparently is prized. People like that blue. It's not quite as simple as alkaline versus acidic. You have to have aluminum in the soil as well, which is a naturally occurring substance. In alkaline soil tends to hold on to that aluminum tightly. Acidic soil, the chemical composition is a little bit different, and so it loosens up its hold on the aluminum, which is absorbed by the plant, and that somehow interacts with the DNA, interacts with the genes that produce the flower color, and make it turn blue. Same genes, different environmental outcome. One of my favorite examples is the Arctic fox, just because they're so cute. This represents phenotype plasticity, where the same animal will turn colors. People are often familiar with the winter coat of the Arctic fox. They blend in well with the snow, so they're less easily seen by their prey. They're less easily seen by the polar bears as they follow the polar bears to get the leavings. If that white coat remained in the summer, it would stand out. So their fur they change fur color from winter to summer and winter again. This is called phenotype plasticity, which just means the phenotype is not set, it's malleable. My eye color is set, it's not plastic, but the fur color in an Arctic fox demonstrates phenotype plasticity. And another example that actually came up in a I've seen this in a couple of AP questions in the multiple choice sections. Some reptiles are not, their sex is not determined by XY chromosomes or by chromosomes at all. It's determined by temperature. So there are sea turtles where if the temperature is low in the hatching or in the growing eggs, you're going to get males. If the temperature is high, you're going to get more females. Did I get that right? Yeah. Conversely, in some lizards, it's backwards. If the temperature is low, you're going to get more females, and if the temperature is higher, you'll get more males. And then there are some other turtles, some land turtles, or freshwater aquatic turtles, where the extremes of the temperature in the growing eggs, the developing eggs, will produce females, and a mid-range temperature produces males. Weird. And there's somehow, what happens is there's an environmental cue early on in development, cold versus hot. If it's cold in this thermosensitive period, then male-specific proteins will be produced. The DNA will be triggered to produce these proteins, which will turn the reproductive material into the testes. Similarly, if it's hot, that's going to turn cause the female. The same genes are going to, same DNA is going to cause be produced. The genes are going to produce female-specific proteins, which will turn that same genetic material into ovaries. It's similar for humans. We are sort of androgynous up until about 6 weeks in the uterus, and then it depends on hormones that's going to cause those same pieces of tissue to turn into either male or female reproductive tissues. In this case, it's based on the environment. Here is a question that came up recently about sex determination by a combination of genetics and environmental temperature. This is an awesome question, and it looks simple, and when you work through it and write it out, it actually works really well. Read it super carefully to figure out what's going on. It's a little more complicated even than the reptiles I showed you. Anyway, final topic. I know there was a question about how do you analyzing pedigrees. I want to be sure we touch on this. So in chromosomal inheritance, one of the things that we want you to be able to do is to explain how chromosomal inheritance generates genetic variation in sexual reproduction. We're going to use pedigrees as a way to understand the pattern of transmission from parent to offspring. So here I've got parents. The box represents a male and the circle represents a female. Very often with pedigrees, we're talking about humans. So the default is kind of just the humans. So these two parents had children. They had you, whoever you are, and they had your older sister. Your older sister married or had children with somebody, and that is your nephew. You would then marry and have a daughter who then marries somebody and has your grandson. So that's just how a pedigree looks. We'll use it to trace the inheritance of an allele through a pedigree. So cystic fibrosis is caused by a single mutation in the CFTR gene on chromosome 7, and it's inherited in a straightforward way. And this is just an example of how some human genetic disorders are due to a single mutated allele. But we can look at this pedigree and figure a lot of stuff out. When there's a darkened circle or square, that is a person who has whatever trait it is you're talking about. So in this case, it's cystic fibrosis. So Laura has cystic fibrosis, and so does Shane. Shane has cystic fibrosis. So that means he's got to have 2 recessive alleles. You already know now something about Rob and Jane, his parents. They each have to have a recessive allele because they each had to give that recessive allele to Shane. They each also have a dominant allele because they're not shaded in. They don't have cystic fibrosis, but it turns out they were both carriers. None of their other children have cystic fibrosis, but we can determine that Paula was a carrier. She inherited one of the cystic fibrosis genes because her daughter ended up with cystic fibrosis, which also meant that Keith also was a carrier. So how can we use this? Let's determine how a trait is inherited based on a pedigree. Is the trait you're talking about, whatever trait it is it inherited as a dominant or a recessive trait? what I suggest you do is just try it and see. If you find even one parent-to-child combination that does not work, then you eliminate that inheritance pattern. You might not be able to prove it is the inheritance you're looking for, at least with a simple pedigree, but you can eliminate some of the others. I'll show you what I mean. Let's take the same pedigree. We're going to try recessive inheritance first. So these two children have whatever trait it is, so they have to be recessive little a, if it's a recessive trait. Okay? So they each got their little a. They each got their little a from a parent. So each parent had to have a little a. Each parent does not have this trait, so each parent has to have a big A. That works so far. And their siblings can inherit a big A from mom or dad, and we don't know what their other allele is, but it doesn't matter as long as they have at least one big A. So could this be a recessively inherited trait? Yes, it works. Let's try it again with a dominant trait. So same children. So now you sort of think backwards from what we normally think. This child has to have a big A in order to have this trait if it's a dominantly inherited trait. So same with this child. Can they inherit a big A from a parent? They can't. The parents have to be little a, because if they were not little a, if a parent had a big A, they'd be shaded in. Since they're not, they're little a, there's no big A's to pass on to a child. So nope, no way for a shaded kid to inherit a dominant allele. That's sort of how you use this. I've put a slide in here you can go back and look at. This it goes into a little bit more detail about how you can eliminate a possible mode of inheritance. And so I would urge you to come back and look at these. I don't want to trace through them right now in detail, but they do show you, for example, this cannot be recessive. Sex linked cannot be confirmed, so you can eliminate some things. Finally, non-disjunction. Non-disjunction is also chromosomal inheritance. It's the failure of chromosomes to fully separate during meiosis. So trisomy 21, or Down syndrome, is a commonly studied example because we know a lot about it. And this is where you get an extra 21st chromosome. Whoops. This happens because there was a mistake during meiosis. So circling back to what we started with, here's meiosis. 4 chromosomes, 2 pairs. Meiosis 1 occurs, yay! In meiosis 2, however, these two sister chromatids failed to separate in time, and they both ended up in the same gamete, which meant the other gamete only got 1 of the chromosomes and it was supposed to get 2. And then over here, everybody's normal, so they're fine. Non-disjunction could occur in meiosis 1 as well. It would be drawn a little bit differently. So what does this mean? This means it depends on what chromosome you got as to what the effect will be. Notice for trisomy 21, that's a small chromosome, and it has a pretty profound effect. Most of us know somebody or know about people with Down syndrome, and it can be a very debilitating condition. It can also be, doesn't affect a person very strongly at all. There's a wide range of effects it could have, which is interesting in and of itself. But it can have a severe effect. And that's a small chromosome. You could have a trisomy of any of these chromosomes. So imagine a trisomy of a bigger pair of chromosomes. That's more genes you have now at play. And the bigger the chromosome, the tendency is the more severe the effects. So having an extra chromosome in humans doesn't work really well. Extra chromosomes in plants is a whole different story. They seem quite happy having extra sets of chromosomes even. Humans, not so much. So there are a few practice questions I've added here at the end. This is one that asks you to think about chromosomes and traits on chromosomes and assorting them. It's a good challenge. I recommend that you give it a try because it makes you really think about what's happening with your chromosomes. Here's one that had to do with fruit flies and talking about the phenotype for eye color. And remembering, this one asks you actually have to remember that the white-eyed fruit fly, it's a sex-linked trait. And one of the purposes of this question was to ask you to recall that, giving you a hint. And a couple more pedigree practices and the explanations that go with them. So you can read about those or cover those up and then see if you can give those a try. So, wow, I want to thank you for watching. I want to say good luck on your reviewing. Good luck on your AP exam. Most importantly, please, please take care of yourself. Please stay safe. And I'm delighted you were able to join me today, and I hope this was useful and helpful. So stay happy, everyone.